Thank you everyone for joining Benchling's webinar on high-throughput DNA sequence design. My name is Ashoka Rajendra. I'm a product manager here at Benchling, and I oversee our molecular biology tools as well as our inventory and registry tools.
Yeah, and I'm Damian Costello. I'm actually a customer success representative at Benchling, and I'm largely involved in the implementation of Benchling 6 applications.
So we're going to start by giving an introduction to Benchling. Who are we and what do we do? We're going to explore some of the trends influencing R&D and specifically molecular biology workflows. We're going to talk about some example use cases for high throughput sequence design, and then Damian's going to walk through a live demo of a specific use case. At the end, we're going to do Q&A. So there's a little button at the bottom of your screen that says Q&A, and if you click into that, you'll be able to submit questions and we can queue those up to answer at the end. To make this a little bit interactive, I want to start with a poll. We're going to be delivering a few of these throughout the presentation just to take a pulse as to what content is going to be important for people to see and hear about. And I want to know, do you currently use Benchling, or if you're not currently a Benchling user? Great, we'll leave this up for about 10 more seconds and then we'll move along. All right, so we have a pretty wide mix of existing users and people who have not used Benchling before. So just a snapshot about Benchling and some of our background. We were founded in 2012, funded by a variety of Silicon Valley venture capital firms. We have over 180 enterprise customers who've been using Benchling over the past year or two, and we have 9 out of the top 25 life sciences companies using Benchling. We have over 99% customer retention, which means that less than 1% of our customers have ever left. We have over 120 employees dedicated to building and supporting our product, half of which who support customers directly or build the product itself. So for those of you who don't know, Benchling has a suite of biologically intelligent applications. We have a molecular biology tool, which we're going to dig into in great detail today, that allows you to do DNA and protein design, as well as a notebook that allows you to capture protocols and record data and results for your experiments. We have a registry system that allows you to not only store your DNA and proteins, but other pieces of data that you would model, such as cell lines, viruses, containers, refrigerators, freezers. We have an inventory tracking system that will track the physical manifestation of all of these samples in your lab. We also have process management tools to manage requests that transit across teams, as well as a workflow tool for high throughput experimentation. We're primarily going to be focusing on the molecular biology tool today. So Benchling's molecular biology tool is a sequence design and visualization tool. So we have a comprehensive suite of tools using new techniques like CRISPR, as well as cloning techniques that are all in an intuitive web interface. We have high throughput capabilities to support your experimentation at scale. And we have collaboration features to allow scientists who are working on designing DNA and proteins in your labs to actually collaborate seamlessly with one another and centralize all this information into the same place. The bedrock of these features is data management. So it is not only important to be able to design all the molecules in your lab, but to centralize all the molecules that are being developed in your lab. So we have a system that allows you to track complex relationship between the entities that you've developed, and we can automatically detect the different constituent parts within sequences. And then these seamlessly integrate not only with Benchling's other applications, but also integrates with your kind of IT environment. So going from left to right, I want to talk about the feature set of Benchling. So specifically our molecular biology tools. So in Benchling, you can do construct design, both at an individual level and in a bulk setting, where you can do restriction-based cloning, Gibson and Golden Gate assembly, and this in bulk and combinatorially. For these sequences that you're designing, we have a primer creation wizard that allows you to create primers if you don't have them for sequencing or qPCR, or even to add regions to your backgrounds and fragments that you need for Gibson or Golden Gate assembly. We also allow you to bring in all of your primers and prior sequences from your DNA and proteins, because we know you're not starting from scratch. So to ensure you're actually using all the molecules and sequences that you already have in your lab. We were one of the first web-based tools for CRISPR, doing guide RNA design, on-target scoring, creation of HR templates, and putting those into plasmids. And we have a sequence alignment tool for DNA and amino acids that can also support up to 100 sequences at the same time, doing template and consensus alignments. Lastly, we have a comprehensive suite of sequence visualization tools. So uploading your own annotations and feature libraries so you can really see the sequences that you're working with and understand what they are. So over 120 of our customers actually use Benchling to support their molecular biology workflows. And what we've learned from these customers is how research and development has been changing over the past decade. There is a major shift in the industry where, on the one hand, you have constant evolution of scientific processes, so new techniques like CRISPR, new cloning techniques, that the software and tools that you use really need to keep up with. At the same time, there is a push to automate rote mechanical parts of scientific experimentation so that science can operate at scale. In order to do that, you need systems that can interface between the software they're using to design experiments and the hardware that's actually executing these experiments. Alongside of this, teams are specializing or going down greater specialization, so there's a lot more collaboration in between groups. And in this drive towards improving speed, productivity, and efficiency, there's also been an explosion of external partners, where large biopharmaceutical companies will often work with CROs, smaller startups might work with large biopharma. So there's a lot of specialization, automation, and scale that requires software systems that can manage this collaboration. So for our second poll, we're curious, which of these challenges do you or your organization face? And we'll give about 20, 30 seconds to answer this. All right. Thanks for all these responses. Really interesting. So moving on to, how do you have software that actually supports the complexity of R&D? You know, on the one hand, the industry is evolving. On the other hand, you're being asked to operate at scale. So a lot of software was designed in a world where it was kind of an individual scientist working on a low throughput capacity, you know, on desktop software where there wasn't a lot of collaboration. So there's a lot of standalone tools that are very powerful and are very good at doing sequence design, but have not evolved to this kind of collaborative, higher throughput setting that scientists now find themselves in. So software has to evolve to account for large collaborative teams, higher throughput design, and be web-based, not just from a convenience perspective, but also so you can integrate with complex IT infrastructure. Biotechnology organizations have no shortage of software, so any new piece of software they use has to integrate well with their ecosystem. So we want to take another quick poll. What tools are you currently using for your molecular biology? Great. Thank you so much. This spans the gamut from Vector NTI, Genius, Snapgene to Benchling. So now I want to drill into what can you use high throughput sequence design for? High throughput sequence design can be used from anything from antibody engineering to CRISPR guide design, where for an antibody, you might need to be generating combinatorially dozens, if not hundreds of antibodies. For CRISPR, you might be designing a similar scale of guides in order to knock out a gene or repair it. In synthetic biology, you might be using this for strain engineering. At an industrial scale, you might be using this to create dozens, if not hundreds or thousands of enzymes. And for plant engineering, designing and knocking out and expressing the right trait in plants also applies to high throughput sequence design. And the point I want to make here is that Benchling has a suite of tools that are oriented towards biotechnology, but they can be used across a variety of different domains. In the demonstration today that Damien's going to dig into, we're going to focus on a subset of our features that are around high throughput sequence design. So we're going to do Gibson assembly, primer design, and primer import, sequence alignment after these sequences have been designed in silico and then brought back into Benchling for confirmation. And then we're also going to show you our visualization tools to how can you bring in your sequences into Benchling, visualize, annotate, and break them up in their constituent parts. So the workflow we're going to talk about end to end today starts with the request for a synthesis and design of a lentiviral construct, which then requires plasmids to be designed and generated by a vector design team. For these plasmids to be annotated and prepared for assembly at the bench, for primers to be both designed and attached for sequencing, and then for sequencing to occur so that you can see, is what I designed in a computer actually what actually was generated in the lab? There are a lot of pain points in this process. You might be doing this workflow end to end, but you might be using several tools to achieve this. Molecular biology tools are often disconnected from your central repositories or central registries of all the sequences that you have in your organization or in your company. Then creating those plasmids at scale versus creating them one by one becomes another challenge. You then have to take what has been designed in a molecular biology application and bring that into lab tools. How do you actually make that transition from what you design in the computer to what is actually assembled at the bench? And then your primer design needs to factor in what do you already have versus what do you plan to design for sequencing. And lastly, the actual sequencing part requires transitioning of files between a sequencing group, it could be a third party, a CRO, and bringing them back in and tying them to the original constructs that you designed. So for another question, I'm curious, which of these challenges do you currently identify with based on the tools that you're using? And that will have Damien as he walks through the demo and to emphasize certain parts of our tool. Great. Thank you so much. So what we're going to demonstrate is going across this entire workflow, how can you do this in Benchling, and how can you use our bulk and high throughput tools to achieve this end to end? So I'm going to transition this to Damien now, who's going to walk through how you can do this workflow in Benchling.
Okay, thank you very much, Ashoka. So I'm now just going to jump into the Benchling platform to begin our demonstration. So before we kind of get into the agenda for the demonstration, just a couple of things that I'd like to address. So the first is that Benchling's a cloud platform for tracking all of the biologics that you work with at the bench. When you're using any of the 6 Benchling applications, you'll notice that I'm actually just in the browser in Google Chrome, for example. So all of the work that you'll be doing is just simply through your browser. You might also notice that I have opened up a Benchling Lab notebook. So the vast majority of today's demonstration will kind of be coordinated within this ELN entry that I've created. Awesome. So let's just briefly take a look at our agenda. So we're going to spend a little bit of time showing you how you can bring in your Vector NTI archive files into Benchling. And then we'll just transition to the bulk of the time, which will be spent on this lentiviral plasmid production demonstration that Ashoka just detailed. So let's begin with this first part, bulk import of Vector NTI files into Benchling. So again, today we're focusing on maybe a Vector NTI archive file such as the MA4 files, but I just want to iterate that you can actually bring in a variety of different file types that are supported in Benchling. And when we mean that they're supported in Benchling, what we're saying is that not only the sequence, but the annotations and tags associated with those sequences will carry over into Benchling. And so it's included but not limited to the file formats that you see here. For today's demonstration, however, I will be bringing in an MA4 archive file from Vector NTI. So you could imagine the typical workflow for this would be, you download or export your archive file from Vector NTI. You then navigate back into your Benchling browser. You find a particular project where you'd like to store your files. You can see that I have a couple of subfolders nested here, including one for today's demonstration. Now, for me to bring these Vector NTI sequence files into Benchling, I can simply click on the Create button and choose Import DNA Sequence. with one simple click and drag motion, I'm able to bring in all 11 of the sequences contained within my archive file into Benchling. So you can see how that literally took seconds to bring in all of these sequences. I just like to take maybe a minute or two to open up a particular sequence to show you some of the display features that Benchling offers within our molecular biology suite. So for example, you'll notice that if I click into a particular sequence, all of the annotations corresponding to the sequence carry over with it. You may also notice that I have my workspace split in half here. So you can actually drag different tabs that are separating your sequences across either side of the workspace. I can toggle on and off different display features, such as the annotations that are present on my sequence map. And any action that I take against one of my sequences directly corresponds or occurs on the corresponding sequence. Benchling's editing features are super intuitive. For example, I can just click directly into a sequence and make an edit, and you'll see that those changes update automatically on the sequence itself. So I highly encourage you to play around with the molecular biology tools if you haven't already. One other kind of cool feature that I'd like to just call out right now is Benchling's version history. So any edit that you make to a sequence can be captured in Benchling's version history, so that if you ever need to revert back to an older version of your sequence, you can do so. Alternatively, you can make a clone or a copy of an older sequence to reference later down the line. Okay. So we can move right along then into the lentiviral plasmid production demonstration. So again, you can imagine that there is a scientist in my group who is going to submit so taking a step back, I am a scientist that's working with the vector design group or vector design team in my lab or institution. And a scientist is going to submit a request for me to design and then synthesize their lentiviral construct at the bench. So to begin with requesting the design and synthesis of a lentiviral construct, you could imagine that the scientist first designs some sort of shRNA oligo. They follow the criteria they need to design these oligos. These oligos will ultimately be used to knock out some target gene in a cell line. You could imagine that the scientist could actually come into Benchling and utilize something like Benchling's registration tables, which is an extension of our registry application, to register the shRNA oligos that they've designed. So with a couple easy clicks, they're able to register these shRNA oligos that they've designed so that they can, reference them later down the line and prevent any redundancy from people recreating those fragments. Using Benchling's request systems, the scientist can actually initiate a request for me to design their lentiviral plasmids directly from the registration table. So for example, they can create a request for a lentiviral plasmid synthesis. They can fill out important metadata like the day they're requesting for this to be designed, the assembly method that they'd like me to use, for example, Gibson assembly. They can even specify things like a vector type that they'd like me to use, for example, the PLK01 backbone. When they create this request, me, the vector design scientist, will receive a notification that this has been assigned to me. I can go and find the proper metadata that I need to do my assembly. And what this does is essentially prevents this kind of disparate system where a scientist has to either email or message or go find another scientist in the lab, ask them to fulfill this request to design this plasmid, hand over the notes that they have, and centralizes it in one place here. Okay, wonderful. So you can imagine the scientist then has registered their shRNA oligos. They've submitted a request using Benchling's request system for me, the designer, to go and build these these plasmids. So before I can go to the bench and actually synthesize the vectors, I need to use Benchling to build my vectors. So in today's demonstration, we'll be using the bulk assembly tool to insert one common promoter in combination with 10 unique shRNA oligos to generate 10 output lentiviral plasmids. And again, we're going to do so using Benchling's bulk assembly tool. So I can first start by navigating to the blue navigational toolbar on the left-hand side to this Create button. From there, I can hover over DNA sequence, and I can actually just choose bulk assemble DNA. This then navigates you to Benchling's bulk assembly tool. The way that the bulk assembly tool works is it's kind of this, like I mentioned, a combinatorial plasmid generator, which allows you to create really complex molecules in bulk with just a couple of simple clicks that are supported by display features and things like that. So I can first start by selecting the fragments that I'd like to assemble. For example, in this case, you might remember that the requesting scientist asked that I use the PLK01 backbone. And then the next step is for me to select a fragment end. Another way that you can think about this is selecting your assembly method. So currently in Benchling, we offer a variety of bulk assembly or assembly methods. Enzyme-based would be, of course, if your fragment was flanked by restriction enzyme cut sites that will produce some sort of overhang. We have homology for quite literally Gibson assemblies, for example, where your fragment has some homology region that's conserved between it and the backbone. And then finally, we have concatenation. So concatenation is an assembly method that allows you to piece together your fragments into a plasmid without it representing any specific technique or experimental technique that you're using at the bench. In this case, the requesting scientist asked that I do a Gibson assembly to design their plasmids. And so I can pick homology as my assembly method. I can then type in my homology regions, for example, TG, 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 GC, 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 GC. You'll see that the last two steps, selecting fragment size and direction, are now supported by display features that appear on the right-hand side of the workspace directly underneath Backbone Summary. I can choose to keep either the larger or the smaller fragment. I can change the orientation of my fragment if I so choose. And again, these last two steps are supported by these display features that we see on the right-hand side. The next step is for me to add my promoter. To do so, I'll click into the Insert One box and follow similar steps to what I just did, navigating through Benchling's tree structure, locating my promoter, entering in my homology regions. And what's fantastic about Benchling's bulk assembly tool is that it automatically remembers the homology regions that we had selected in the previous window. So I can simply click on my previous homology regions. I can add in new homology regions if need be. And again, you can see that this last two steps, fragment size and direction, are supported by these display features on the right-hand side. The last step is for me to select the shRNA oligos that the requesting scientist had built. So I can easily select all of the fragments in my folder in bulk by simply clicking on the folder to highlight all of them. Alternatively, I can simply shift select the fragments that I'd like to assemble to very quickly and efficiently highlight these fragments. One final time, I can choose homology. I can pick my particular homology regions. Benchling sanity checks me on the right-hand side, assuring that I've selected the correct fragments. My last step then is to choose a folder in which I'd like to save my recently designed plasmids to. For example, my assembled lentiviral plasmids folder. I could always create new folders if I need to. And because Benchling's molecular biology suite is integrated with all of the other applications, I'm actually able to select a schema, for example, lentiviral plasmids, so that I can automatically assign any critical metadata that I need to capture against my plasmid to these plasmids as I design them, right? So benchling is now prompting me to take a bulk action to build my lentiviral plasmids, and then allowing me to automatically capture any data that I need to against these plasmids using Benchling's registry system. I can now assemble my plasmids. When I click on the open button, you'll see that all 10 of my assembled plasmids appear. So in a matter of seconds, I've now created 10 unique plasmids, each containing the same promoter and a combination of one of my 10 shRNA oligos. If I click into a particular plasmid and jump down to the 5' UTR, we'll see that my insert sequence, in this case my promoter, appears here, followed by my insert sequence. However, as a vector design scientist, I need to be able to jump into a particular plasmid and quickly reference the components or the fragments that are comprised within my plasmid for future reference when I'm doing other assays. In other words, I need a way for myself to tag any of the sequences or parts in my plasmid for future reference. Now, I could go through, I could highlight a particular sequence, I could right-click and create an annotation for my promoter for each one of the fragments that I've created, but I don't want to have to do that for all 10 of the plasmids I made. And in your case, you might be designing 60, 80, 100 plasmids all at once. You can see how this would be very time-consuming. So what I'll now do is I'll leverage Benchling's bulk auto-annotate function to apply tags to all 10 of my sequences. To do so, I can simply click on the checkbox above all of my sequences to highlight them. Navigate over to the More dropdown, where I'll have an option to analyze my sequences. And here is a wonderful moment to stop and show you all of the unique bulk actions that you can take against a vector that you've designed. In this case, we'll be auto-annotating our sequences. I can choose from a library of features or tags that I've previously created. For example, my shRNA library. I can auto-annotate my sequences. And in less than a second, if I click back into one of my sequences, you'll now see downstream of my 5' UTR is my promoter, followed by the shRNA fragment that I assembled, the homology regions that I used to assemble them, etc. And the same goes, for example, for another fragment. If I open this here, we'll see the same promoter that we saw in in plasmid 1 rather, followed by the next shRNA oligo that my requesting scientist has built. So in maybe 6 minutes, I was able to bulk assemble 10 very unique plasmids and automatically set or establish the metadata I need to capture against them and tag the sequence with all of the relevant parts. Again, you keep hearing this reoccurring theme that Benchling's molecular biology suite is integrated with other applications. If I wanted to take my tagging a step further and create links between the vector that I've designed and its constitutive parts, I can use Benchling's autofill parts function to, for example, associate the promoter that I use to build these particular vectors. Benchling will then autofill the parts and actually create a link to the promoter that I used in the assembly, where I can find things such as its sequence map, and I can come into the registry and capture any metadata or any important information against the promoter itself. So now that I've designed my vectors, my theoretical vectors, you could say, I've annotated them or tagged them so that I can reference the pieces at a later point. The next step is for me to actually go to the bench and synthesize these vectors, right? So what I have displayed here for you is an assembly protocol, right? In this case, as the vector design scientist, I might have already created a PCR fragment or a fragment that contains the homology regions that I used in my Gibson assembly. I can actually mention those fragments here to create links to them so that I can reference them later down the line. You'll see that when I click into this, I can see any relevant information about the primer that I designed in Benchling's primer tool, as well as all of the particular fragments or the particular vectors that this has been attached to in the past. So quickly referencing the primer and its associated plasmids is as easy as clicking into the @mentioned primer. I then might do something like run a 2% agarose gel and gel extract my DNA fragments. If I've taken an image of my gel, I can attach that into Benchling's notebook here. I can go back to the bench and combine my fragments, following some sort of standard Gibson protocol, for example, combining my fragments in equimolar concentrations. Take the next steps to do a classic transformation of my plasmid DNA in E. coli, such as DH5 alpha. I can plate that on an agar plate, pick my colonies. I can amplify them in something like LB broth, and then ultimately use some sort of kit, for example, to obtain the plasmid DNA associated with the lentiviral construct that I designed. In similar fashion, you could imagine as me being a plasmid vector design scientist, I might want to register these plasmids now that I've gone to the bench. I've actually created them. I've captured specific data against them. I might want to, again, leverage Benchling's registration system to capture and monitor these vectors down the line. Actually, Benchling's inventory application integrates with the molecular biology suite and the registry as well. So I can use something such as a transfer table, which is an extension of our inventory application, to actually directly add the samples that I've recently created, for example, the plasmid entities that I've recently created, and transfer them automatically to, for example, an Eppendorf tube, which can live within a box, so that if I need to go quickly find these plasmids to resubmit for sequencing or what have you, I can find immediately where they are. This is also particularly useful if the scientist wanted to go back and tell the requester, what the plasmid was that I designed and where it's stored. They can actually reference the same information from the notebook entry that we're collaborating on. Okay, great. So now I've done this. My plasmids have been mini prepped. I've maybe made aliquots of them. I've put them in some sort of box in my -20 or my -80. The next step for me is to then, attach maybe some primers that I've created in the past and design additional primers that I would need to get additional coverage before I submit my samples for sequencing, right? And so again, I don't want to have to go through and attach all of the primers that I've already created in the past. Maybe my backbone has some common promoter that I've designed primers for sequencing in the past, like the U6 promoter, for example, and the PLK01 backbone. So I want to apply these primers in bulk. So again, I can take similar steps to those that I took before. I can select my backbones. Again, come over to the More and Analyze functions. I can attach my primers, and I can pull these primers from a folder that I've saved them to in the past, right? So again, this prevents redundancy. It prevents you from recreating primers that you've already made in the past. Nobody wants to have 8 variations of a U6 primer or promoter primer. I can attach these primers to a particular sequence. By the time I finish my sentence, all 10 of my fragments have the primers attached to them. Again, this is particularly useful if you guys are designing like 100 plasmids, for example. You don't want to attach the same primer to 100 plasmids individually. If I actually click into one of the particular sequences that I've created, we can see that this plasmid, or that these primers are attached here. If I was a thorough scientist and I wanted to make sure that I had primer coverage across my entire sequence, I could use Benchling's primer design tool, for example, our primer wizard, to set extremely granular parameters around my primer design. And I can do it very quickly by using the selections that prompt me to pick a qPCR primer versus a sequencing primer. I could even get into some really gnarly, melting temperature parameters if I wanted to. For example, the molarity of my DNTPs. I can set criteria around minimal and maximum GC content, melting temperature, primer size. I can tell Benchling to create primers for me in bulk across the entire sequence by telling them the number of sequencing primers that I'd like to build. Ultimately generate my primers. Benchling will go through, find the criteria that I've established, and create not one, not two, but in this case, 6 different primer pairs that I could use to attach to my sequence. In this case, I highlight all of them in bulk, assuming that I want to keep all of these primers. I can save them to a folder, for example, the shRNA oligo sequencing primers folder that I previously pulled from to attach my primers. And again, this centralizes the primers that we're designing through Benchling's molecular biology suite and prevents rework in primer design. Awesome. So now I've designed these primers in Benchling. Again, you could imagine I might go back to the bench. I now, make my mixes of primers and mini-prep DNA following the instructions for the CRO that I'm submitting this to sequencing to. I prep my samples, I send them to sequencing. I patiently wait for my sample data or my sequencing data to return to me. And then I can bring these AB1 files, for example, back into Benchling. Now, there's 2 ways that you could typically do this very quickly. The first way would be to, import your zip file or your AB1s via the import function that we use during the Vector NTI archive file import. Alternatively, and to popular demand, you could use Benchling's API to actually push your sequencing results directly into a particular project so that you don't have to even leave the Benchling platform to collect your data. The last step for me is to kind of QC. I want to confirm that the theoretical lentiviral vectors that I designed using Benchling's bulk assembly tool match what I did at the bench. And so what I might do is bring my particular sequences into Benchling. You can see, for example, I have a nested folder for sequencing results. Again, I can click on this expanded window to create my alignment. Now, in this case, I went and picked 3 colonies from my agar plate. I submitted them to sequencing, and now that I'm aligning them to the vector that I originally designed in the bulk assembly tool. But you could imagine doing this, especially if you're like picking colonies or you have a lot of AB1 files, you can imagine doing this for up to 100 sequences at once, creating an alignment very quickly. So one more time, come over to the Analyze tool. I'll go ahead and pick Create DNA Alignment. You'll see that Benchling automatically grabs all of the sequences in this particular folder, hoists them into the alignment pop-up panel. I have an option to add even more sequences if I forgot to do it. Maybe I have some saved on my computer. Maybe I have some in Benchling already that I want to search for and add. And maybe I want to just go ahead and create a new sequence, give it a name, and type in the bases. So you have an opportunity to add more alignment or more sequences in your alignment in bulk directly from the pop-up window without having to navigate out of the alignment tool. Choosing between a consensus alignment and a template alignment is as simple and easy as clicking on the fragment that you'd like to designate your template. The last step then would be to create your alignment. Benchling will go through, understand which sequence that you've made as your template, will create the alignment for that particular template, and save that alignment to the template sequence. So you'll see here that we've now created one of our alignments, and you might notice that there's a lot going on here, and this is because our alignment tool offers an abundance of very, very interesting, unique display features. all of which can be toggled via display icon, similar to when you're looking or viewing a sequence map itself. I can look at specific reading frames. For example, I only want to look at one reading frame. I can drag across this mini map here to find any of the mismatches that appear in red or any ambiguous bases that appear in yellow. I can even show a compact view if I'm working with a large number of sequences and I kind of want to just see the mutations, for example, or the differences between my AB1 files and my template alignment. And so you can imagine doing something for a much larger alignment, something that might look like this, where you're aligning 30, 40, 50, 60 sequences in one alignment. And this allows you a more global view of the samples that you prepped on the bench compared to the ones that you've designed in Benchling. So you can see now in a matter of 28 minutes, I was able to design a number of plasmids based off of requests that a scientist has made to me. I was able to annotate or tag my sequences with the components that are, within my particular plasmid. I was able to promote those fragments to the registry where I could capture critical metadata against it, seamlessly attach sequencing primers that I had designed in the past to the vector that I've created, design new primers in bulk against a particular sequence, bring my AB1 files in and create an alignment without ever having to leave the Benchling platform, and in doing it just, quite frankly, very quickly, right? So I'm now going to hand it back over to Ashoka, who is going to kind of talk about what sets Benchling apart from our competitors, and then we'll open the floor up to some question and answers and wrap things up here.
Thank you, everyone. And I've been keeping track of a lot of the questions that have been coming in, so we'll kind of go through them one by one in a moment here. So just to kind of summarize, we have a workflow where, you start with a team who wants to design a construct and needs to do that in bulk, needs that to be assembled at the bench and have it be sequenced to confirm it, and then so that it's actually able to be used in the lab. And Benchling has tools to facilitate each part of this process. So we can help you with your requests. We can integrate with your registries to find the parts that you have. You can bring in the data from the files that you have already on your computers. We have tools that allow you to design the sequences that you need. We can then annotate these things, bring them into a physical representation when you actually use them at the bench, and then connect that to the sequencing data that you ultimately generate. So kind of Benchling at its core is built with the scientists in mind. If scientists don't like our tool, then we're not doing our job. It's also built to integrate to your complex IT environment. We're built for insights insofar as, you can track anything that ever occurs to the molecules that you work with, ranging from their design to where they came from, to how they're used in assay and screening, to their sequencing results, all the way down throughout the process. And we have a customer success team who works with you to actually map your science to the solution. So with that, I'm going to go through some questions here and some Q&A. If you'd like to dig in more, if you're not currently a user of Benchling, you can reach out to us and we can demo this functionality more extensively. I know we covered a ton of material here, and the goal here is for you to understand kind of what's possible, and we can work with you to understand your specific scientific workflow. And if you're an existing user, feel free to reach out to your customer success representative. So I'm going to go through here kind of in chronological order to all the questions. There are quite a few. And some are in the Q&A window and some are in the chat. So I'm going to start to the top. So one of the first questions was, does Benchling have an API? And that's a great question. So we showed bringing in data from files, and that's often the case. People are exporting them from design tools that they use. And we also have an API to allow you to upload all the sequences that you work with. The rest of this answer is going to actually answer a couple other questions. So some people asked, can you create 300 alignments for 100 templates, or how do I bring in my libraries and proteins? So when companies are operating at scale, or if they have their own registration system, what they often do is sync that with Benchling. So there's a kind of two-way communication between what's in their registration system, whether it's fragments or parts or primers or what have you, to be pushed into Benchling, and then what's generated in Benchling to be pushed back into their own core system. Another thing that this helps facilitate is that if you're doing a lot of sequencing, again, you don't want people to be shopping around with files. So what is commonly the case is sequencing groups can, using our API, automatically create the alignments inside of Benchling. So the scientists can just go back to the sequence that they assembled and actually create it, or actually view it themselves without having to worry about where it came from. Let me scroll down. So sorry, there's another question. Are sequences stored in my computer or dragged from a database, or for example, NCBI? So just to show you a quick back, kind of similar to the API question. So you can import files directly. They could come from Snapgene or Vector NTI or SeqBuilder, what have you, Genius. You can search external databases like NCBI and quite a few other publicly available databases by searching gene name. You can just specify an entire chromosomal region, or you can paste in a raw sequence. And in addition to that, you can also, again, use our API to synchronize this with an internal system that you might have. So is the request mechanism a critical prerequisite of using the bulk assembly tool or in silico cloning? No, it is not. So what we showed you today was primarily our molecular biology tool to do this high throughput sequence design. We also showed some of the other applications like our requests in our notebook. We want to emphasize the purpose of showing them is to show kind of a way that you could do that, but it is often the case that people have perhaps their own request system. So Benchling, being a cloud platform, is flexible to perhaps a request system might have to interact with Benchling that you have built or you have purchased. So it is in no way a prerequisite, but simply showing how you might send something from one team to another to fulfill.
Yep, and then just to add a little bit to that too, you can think of it in the sense that a lot of the molecular biology tools that we just demonstrated will save an abundance of time, especially when creating molecules in bulk and doing some sort of action against those molecules. And our applications such as requests, inventory, and registry kind of add a second layer in which you could save extra time. But certainly using the molecular biology tools alone will save you an abundance of time.
Mm-hmm. So Damien, there was a couple questions about schemas, and did you want to kind of show what setting basically what is the purpose of setting a schema, and how does that allow people to actually have complex data models beyond the sequence itself?
Sure, and I'd just like to call out too that to keep in mind that a schema is actually a concept involved with the registry itself. And so, for example, if I just jump into one of my sequences here, you'll notice that under this metadata tab corresponding to this sequence, we will have something that we call a schema. So in this case, my schema is a lentiviral plasmid. And when I designate something as a lentiviral plasmid schema, what I'm doing is associating certain data points that I need to capture against all lentiviral plasmids. And by selecting lentiviral plasmid as my schema or my sample type, I get these associated data fields that I can fill out manually by clicking on this edit button, or by leveraging something such as Benchling's autofill parts to automatically create a link between the part that I use to design my vector and the vector itself. So definitely a registry concept there that is worth leveraging if you are going to be using the molecular biology tools heavily.
Yep. So scrolling through, there are quite a few questions. I'm just trying to find one that kind of answers a lot of them at the same time. Is there a way to sequence data alignments for the 10 different plasmids in QC in one go? So that's another one where, again, you can do this manually. We recommend using an API that will kind of programmatically create all the alignments. Another question, is codon optimization available? So that's a feature we're working on this quarter, and we expect to release it at the beginning of August. So if you're an existing user of Benchling, stay tuned for that. We'll be reaching out to you to try out this feature so you can use this for optimizing sequences based on organisms as well as back translation from proteins. And that will also kind of go along with codon frequency tables. And let's see.
Yep.
Is it or go ahead?
Yeah, I saw one that said, can we build our own cloning workflows? It's actually an excellent question and something that our users do quite frequently. Oftentimes what we'll see is they'll create some sort of cloning protocol or standardized, experimental protocol in Benchling's lab notebook. And then they'll use the bulk assembly tool to actually clone their vectors. Many folks will actually leverage our concatenation method to design their vectors using some of the other cloning methods that we didn't explicitly describe via, for example, restriction enzymes and Gibson, and then actually create specific primers to make those fragments on the bench. So that's certainly something that our clients do often. And if you're working with a customer success representative, they can certainly help you in designing and kind of optimizing that workflow.
Great. So there are a few questions that relate to actually features on our near-term product roadmap. So there is one question about, can you generate work lists for robots? So kind of to zoom out on that question, we work with many customers who they might start with creating dozens, if not hundreds or more sequences of proteins they ultimately want to express and run through screening and sequencing and so on. And the natural kind of endpoint for this is, if you're doing this at scale, is to use this in liquid handler automation. So we're actually currently working on that feature. Over the course of the next 3 to 4 months, we'll have an initial release of this. So if you're an existing customer of Benchling, please reach out, or if you're not, and we can talk about our plans there, because that is in progress with several of our key customers to set that up. There's another question: can you order oligos guides, dsRNA from synthesis companies like ADT? Yeah, so there's no kind of like native ordering button inside of Benchling, but what is commonly the case is people will design constructs inside of Benchling and then go to a synthesis provider like IDT and order them.
Yep, and Benchling in the molecular biology suite offers a variety of export functions. So if you ever wanted to export your primers, for example, to a CSV file and then copy those over into IDT, you can do so pretty easily.
Yep. And there were a couple questions about and related to kind of import formats and export formats. So, if you're exporting your files from Vector NTI or Genius or kind of other tools, will they be recognized natively? The short answer is yes. Particularly if you're using kind of open file formats like GenBank, which are kind of a standard format. And what we've built are importers for kind of application-specific file formats like Vector NTI and Genius. So in the kind of rollout process, what we'll do is kind of load in all the files, and if there's any issues with kind of annotation showing up, then we'll modify. But we think we have a pretty good solution for just dragging and dropping files straight into Benchling. Scrolling through here to more questions. So can you search? Yes, what specific search would you like to do?
Yeah, we can just actually take a second to talk about search in general. So again, I'll just kind of jump back into the platform here. So Benchling actually has a pretty powerful search functionality. So search can be accomplished through 3 methods. You can search within a particular project or subfolder within that project if you need to. If you have a registry, you can actually come into a particular registry and search for things within the registry itself. Or you can actually, and I would recommend using Benchling's like global search option, which allows you to essentially look for a particular entity. For example, I might want to find like a particular guide. I can then apply folders or filters if I wanted to. For example, I could look for something such as the author that created something. In this case, I could search for Shaw McCormick or Stefan, for example, somebody who already exists in my organization. I can add more granular filters on top of that. For example, I can search for the date that something was created. If I'm using the registry, I can look through particular data points. So we have a variety of ways to search and filter through the UI. A lot of folks will actually take advantage of the expanded view as well. For example, if they want to find a particular search for a particular item and kind of get a global look at what's going on there. So I highly recommend that you use the global search function when interacting with the UI.
Would tags associated with specific schema be queryable by global search? Yes.
That's another question here.
Any tags associated. So any like schema field. So a schema, just as a reminder, as a concept, is kind of a data model. So you might start with the sequence, but for a given sequence, you might want to know, which part is the promoter, which part is the backbone, which part are the inserts, et cetera, et cetera. You could define other fields like, state and status, like this sequence is designed in silico or the sequence has been synthesized. But any kind of additional kind of field that you put is all searchable and queryable by our global search.
You can also use like a control or command search when you're looking at a particular sequence. So you might notice that in the upper right-hand corner here, I can now search for specific bases, for example, GGG. Benchling will highlight those on the sequence map. I can also search for a particular annotation, such as the HIV-1 annotation. So you can search within an individual sequence as well. So it looks like we got about 1 or 2 minutes here, maybe 1 or 2 questions before we wrap things up.
Yeah, so I'll answer last couple of questions. So can you automatically detect homology? That's something we're working on over the next 3 months. So, basically taking your constructs and finding kind of those flanking regions automatically for you to kind of speed up the process of assembly. Another important one, will this webinar be available later on? Yes, so we are recording this webinar and we'll send that out to all of you. We covered a lot of material and, if you want to dig in further, again, I think, reaching out to us to kind of tailor kind of a demonstration to your needs works best. So we're going to close with one final poll. And just share your feedback. If this was interesting to you or helpful, please let us know. We want to do more webinars in the future. We want to tailor them to different audiences. So really appreciate you taking the time and please give us your feedback on how it went.